This is the Asian Organization of Clinical Genetics presented by the SchrDbb. We reference ABC of Clinical Genetics by Helen Kingston. So here we're going to visualize first the issue of clinical genetics. Now you need to know that the main pioneer inside clinical genetics are Gregor Mendel. So Gregor Mendel has worked between 1822 to 1884, and he's the founder of the Mendelian theorem. Now, the Mendelian theorem of clinical genetics is a basic theorem in genetics. Why? Because it speaks about the Mendelian theorem of clinical assortment. Are you getting? So we have two main theorems which that was found by Gregor Mendel. The first theorem is that of the genetic spread. That is when the father and the mother, the father and the maternal genes can be assigned to the different daughter cells. And now the second theorem was the theorem of assortment. So all that were used by Gregor Mendel. Now the second thing, you have another person that was also a very good pioneer in clinical genetics, who was also Archibald Goward in 1858 to 1936. So he also worked in order to improve the clinical genetics model. So these are the people that discovered now the DNA structure. The DNA structure was discovered by James Watson and Francis Crick. Those are the two people that discovered the DNA structure. Because actually in the past it was thought that it was difficult to know that the genetic material of living organism was stored inside the DNA. So there was a research that was made in order to know where the genetic material was stored in. Is it clear? Because you need to know that the chromosome both contain the histone protein and the DNA. Some people were thinking that the genetic material are inside the histone protein because proteins have a very high level of organization. The tertiary structure of protein can be changed from one form to another. So people were mainly thinking that the genetic material of a living organism is contained inside the histone protein. But Watson and Francis Crick came out with the DNA structure, and then they found that the genetic material of the living organism is inside the DNA and not inside the protein. So that is a brief issue. Now you need to know that genetic diseases generally, the genetic diseases you are going to have different types of genetic diseases. You have the single gene diseases. Second one, you have chromosomal abnormalities. Third one, you have common disorder with appreciable genetic component. You have congenital malformation. So all these are newborn diseases. So when a newborn is born, when you have a newborn, the first thing that you can think of, you have single gene disease. Second is a chromosomal abnormality. You also have even multiple gene diseases, where multiple genes are involved in the production of that disease. You have chromosomal abnormalities, you have common disorder appreciable genetic components, and then you have congenital malformations. Now, single gene disease is the basis of is going to use the basis of Mendelian theorem of clinical assortment. Is it clear? So when you have this, so that single gene disease is divided mainly into three, which are autosomal dominant diseases, you have autosomal recessive disease, and you have X-linked disease. But now, in a prevalence of 1,000 population, you see that the one which has congenital malformation are the most prevalent. Example, you can have just cleft lip, club foot, cleft palate. So those are congenital malformation which do not have any genetic component. So they may just be formed because of malformation of the organ when during the embryologic period. Is it clear? Now after that, the second one, which is also very important, is a common disorder with appreciable genetic component. We have other disorders which have clinical syndromes like Down syndrome, but Down syndrome is more of a chromosomal abnormality than a common disorder. So chromosomal abnormality is why you see that is 6 to 7. It does not really have a genetic component. Down syndrome only have a genetic component provided it is produced from a translocation that was inside the mother. Example, a translocation between chromosome 14 and chromosome 21, as we are going to see later. So we are also have now after the chromosomal abnormality, we have now autosomal dominant disorders. You see that X-linked diseases and autosomal recessive diseases are not very common in our context. So they are not very common. The next thing that you have to note is the type of genetic diseases. So the type of genetic disease, as I've initially said, you have single gene disease which respect the Mendelian principle. You have multifactorial disease. Multifactorial diseases are diseases that both depends on the gene and the environment. Is it clear? It depends both on the gene and the environment. So that is why it's called multifactorial, because you need to know that the phenotype is equals to genotype times the environment. So that's a multifactorial disease. Now chromosomal abnormality is rare, as you have seen the clinical prevalence, it's rare. And we said that it is involving the growth structure of the chromosome. That's a chromosomal abnormality. And then we have somatic mutation. Somatic mutation is not actually having a genetic component. It is mostly because of when there is cleavage, there is a malformation, an abnormal mitosis or non-disjunction which occur within the mitosis of the cells that are produced at the level of the zygote. Is it clear? So all the lineage of that particular cell is going to have a disorder. So that's a somatic mutation. Now let's visualize now the organization of genetic services. Now we need to know that in the organization of genetic services, you have what is called clinical genetics. Clinical genetics is just a service which is involved with, number one, it is involved with diagnosing genetic diseases. So clinical genetic services are involved with one. Number one is involved with diagnosing genetic diseases. The second thing, because you need to know that genetic diseases are not easily treatable. So what they are going to mostly do is to prevent genetic diseases from occurring. They diagnose genetic diseases either after birth or before birth. Is it clear? Or even before marriage. Is it clear? So those are diagnosing clinical before or after birth, diagnosing clinical genetic diseases. The second thing is to prevent the reoccurrence of genetic diseases. And to prevent the reoccurrence of these genetic diseases, we have now to use different set of instruments to help us in that. So now, so you can see this is a list of common reasons for referral to genetic services. We have children with congenital abnormalities, that's diagnosing genetic diseases. Though congenital abnormality is not a specific genetic disease, can be considered as one. So when you have any congenital abnormality like disability, default, dysmorphic features, it is a part of that. So diagnosing children with chromosomal disorder or inherited, diagnosing adult affected with congenital abnormality, diagnosing adult known to carry a risk of carrying. Now this one is to prevent the reoccurrence. So if you have an adult known to carry a particular trait of a disease, it is now, you are now going to try to prevent the reoccurrence of disease in the future generation. Because when a child is affected with a particular disease, there are certain set of questions that the parents are going to ask you, that a clinical that's the main goal of clinical genetic service, such that as to answer all these questions. The first thing is, why is it that my child is the one that have it? So that's the question that clinical genetic service can help you to answer. The second question that parents are going to ask you is, how can the child live with this clinical disease, with this genetic disease? That's the second question that genetic service can answer you. The third thing is, how can I prevent the reoccurrence of this disease such that my other children don't have it, or my family members don't have it? So all these are the questions that are going to be answered by clinical genetic services, which are usually asked by parents. Now we need to know that what in clinical genetics there are different components that can now be used now in order for you to prevent the reoccurrence. Because in order for you to prevent the reoccurrence, let's imagine that the disease has a genetic component. You need to diagnose if both parents have any genetic abnormality. Now, to diagnose if a parent has genetic abnormality, you can use cytogenetics tool, or you can use molecular genetics tool. Now, in cytogenetics tool, we have example like karyotyping. So when you karyotype, you are going to have an array of the 23 pair of chromosomes that you can see both of the picture of the mother and the picture of the father. And that karyotyping can help you to know the gross morphology of the chromosome, if there is any abnormality with the gross morphology of the chromosome. Now, still in cytogenetics, we have also what is called the fluorescein in situ hybridization method. And that fluorescein in situ hybridization method, let's say that the child has a Down syndrome and you want to rule out the fact that the Down syndrome comes from a translocation. You are going to use the fluorescein in situ hybridization for the part that is involved with the Down syndrome. Is it clear? So when you have that in situ and then you visualize that there's a disorder, it can help you also in cytogenetics method. Now the next one is molecular genetics. Now for molecular genetics, what you're going to use now, we have different aspects, like even in cytogenetics we have computed array distributions. So those are the different things which are involved there. Now in molecular genetics, here you need to know that mostly this genetic part is going to be involved with the DNA test. So here you are going to visualize, because you need to know that there are certain biochemical diseases that occur in certain patients, like inborn error of metabolism, which can be diagnosed at the level of molecular genetics. So these are some common reasons for molecular genetics analysis. Is it clear? So the first one, we have cystic fibrosis, hemoglobinopathy, Duchenne and Becker muscular dystrophy, and all that. So you can just pause the video and visualize all that. And then the next one, we have biochemical genetics. So I just made an error now. Molecular genetics is in order to involve an abnormal molecule. Is it clear? Because you need to know that what certain things like cystic fibrosis, there are certain particular proteins that the patient has cystic fibrosis are going to lack. Another thing like Duchenne and Becker muscular dystrophy, you need to know that they are mostly involved with the dystrophin protein and the dystrophin gene. So that particular molecule is lacking. So that's why you are calling it molecular genetics, because you are trying to find the molecule which the patient is having the disorder in. Like other myotonic dystrophy, the patient is going to lack myosin filaments. So this patient now, you are going to try to find if there is enough myosin filaments inside the muscle. So if there isn't, you see that is going to be involved in molecular genetics, and there's a particular gene that is involved in the production of that particular molecule. That's why it's called molecular genetics. Now the next genetics that you can study now, that I made an error before, is biochemical genetics. Biochemical genetics is mostly involved now with inborn error of metabolism. Is it clear? So it's a specialized biochemical department, genetics department, that offer clinical laboratory services for a range of inherited metabolic disorder mostly. So example of inherited metabolic disorder, we have phenylketonuria, we have congenital hypothyroidism. So those are the different things to be seen here. Now the genetic register and all that is mostly involved now with people that are going to specialize more in clinical genetics, so we are not going to speak about that. So for me, we say thanks for your kind attention, and don't forget to like and subscribe for our channel. Thanks very much.